So is it going public?
We are live on Instagram.
Okay. So Bismillahirrahmanirrahim. Allahumma salli ala Sayyidina Muhammad wa ala ali Muhammad sallallahu alayhi wa sallam. So it's a real pleasure to introduce Professor Dr. Amir Faisal, who is a faculty in Department of Life Sciences. And he is going to explain us what is behind the Nobel Prize for Physiology and Medicine in 2023, which was given to Katalin, Karikó, and Weissman. Faisal is, it's also a pleasure to me that I'm introducing Faisal. So Faisal is actually a highly accomplished fellow at School of Science and Engineering. He did his PhD from Friedrich Miescher Institute in cell biology, biochemistry. His expertise is in cell signaling. After his PhD in 2003-04, he moved for postdoc in Peter Parker's lab in Cancer Research UK, I believe. And then after his postdoc stint with Peter Parker, he joined Institute of Cancer Research, where he spent nearly 10 years in advanced anti-cancer drug discovery labs. And some of his discoveries, they are already in advanced clinical trials. And since last 10 years or more, he is at SSE, School of Science and Engineering, where he has established his own research group working on cell signaling, as well as a major contribution is establishing early stage drug discovery here in Pakistan. So over to you, Professor. Thank you very much.
Thank you, Dr. Tariq, for very generous introduction. All right, I'll start in the meantime anyway. So the talk about how this Nobel Prize was given to these two scientists who have actually discovered or innovated the use of mRNA, and we've seen the vaccine development during the COVID-19 pandemic. But there are many other applications, and people have been trying to use mRNA for many years. So their discovery actually enabled the use of mRNA for all those plethora of applications in different fields of therapeutics. So the talk actually is, I've designed it to be, as it says, accessible talk. So it's going to be very basic. But looking at the audience, I think I can go into some detail when I go further. So this is kind of a brief overview of what I'll do. I'll obviously introduce you to nucleic acids, mRNA and proteins. We'll talk about what are the challenges, or what the challenges to use mRNA as a therapy, and then what was actually the breakthrough. There are 3 key papers listed on the the website for the Nobel Prize that enlist the key discoveries, and I'll talk about those 3 papers. What is the take-home message from those 3 papers? What's so significant in those papers that earned them the Nobel Prize? And then at the end, I'll briefly talk about applications other than the vaccines. So, All right, so as I said, this is supposed to be very introductory. So when we talk about life, we talk about how we define life is that, things that share common characteristics, we call them life. And one of those common characteristics, actually a unifying characteristics in all of life, is the chemical composition. So 98% of the living organisms on this planet actually are composed of, or all the living organisms on this planet, 98% of them is composed of 6 elements. It's carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Right? So, and the rest 2% is different other elements that are very important, but majority of organisms, they share these 6, actually all of the organisms, they share these 6 important chemicals. So when we look at the chemical composition of living organism, majority of us is actually water. And then about 30% is macromolecule. A macromolecule is a molecule that has a molecular weight of greater than 1000. And there are 4 major types of macromolecules. There are proteins, there are nucleic acids, there are lipids, and then there are carbohydrates. So for today's talk, our focus will be on proteins and nucleic acids. 3 of these macromolecules that I just mentioned, they are actually polymers of smaller units. So they are covalently linked smaller units that make a long chain. For example, proteins are polymers of 20 different amino acids. Similarly, nucleic acids are polymers of nucleotides. Right? So we'll talk about these nucleic acids and proteins in the next few slides. So nucleic acids, as I mentioned, are polymers of nucleotides. A nucleotide contains 3 important things. It has a sugar, it's a ribose or deoxyribose sugar depending on the nucleic acid. It has a nitrogenous base, and it has a phosphate group. And these nucleotides, they are linked together by a chemical bond. So, for example, another nucleotide can link via covalent bond to the previous nucleotide. So you have these nucleotides linked together. So when we have this polypeptide chain when we have this polynucleotide chain, this polynucleotide chain has one end that has a 5 carbon that is free. So here there is a carbon that is carbon number 5, 1, 2, 3, 4, and 5. And the other end has 1, 2, 3, 4, and 5. Other end has carbon number 3 that is free. So when we have a polynucleotide chain, we call it कि उसका एक end जो है that is a 5 prime end, the other end is a 3 prime end. So we have these nitrogenous bases. There are 4 nitrogenous bases in DNA: guanine, adenine, cytosine, and thymine. And so there are 4 different types of nucleotides: adenosine, guanosine, cytosine, and thymine. And these nucleotides, 4 of them, they make the whole DNA. So the polynucleotide chain is sequence of nucleotides that are composed of these 4 different nucleotides. Now, when we have these nucleotides attached together, as I said, this can be directional. So we can say it goes from 5 prime end, which is this one, and it grows in the other direction, which is the 3 prime end. So the new nucleotide will always be added on this side. I'm not going into detail to why is that, and then because DNA is a double-stranded structure, so you have 2 of these polynucleotide chains that run anti-parallel to each other. So one goes 5 prime to 3 prime in this direction, the other goes 5 prime to 3 prime in the other direction. Now one of the properties of these nucleotides is that they some of them, they are complementary to each other. Complementary means they form hydrogen bonds with the other nucleotide. For example, guanosine makes a hydrogen bond with cytosine. Adenosine makes a hydrogen bond with thymine. So these are, there are large double cyclic bases, we call them purines, and the single ringed structure we call them pyrimidines. So the double stranded structure of the DNA, the two strands of the DNA run anti-parallel to each other, one in one direction, the other one goes in the other direction. And the bases, they make hydrogen bonds with each other. These hydrogen bonds are one of the weaker bonds in chemistry, right? They are not as strong as the covalent bonds, but there are so many hydrogen bonds in the DNA, there are so many nucleotides that these two strands of the DNA, they are held together by the force of these large number of hydrogen bonds. So within, C and G, there are 3 hydrogen bonds. Within A and T, there are 2 hydrogen bonds. And then this this ladder-like structure is turned into a helix because of the structure of these bases. And this helical structure that was discovered in 1953, and that structure, actually one of the most important things in that structure was it gave an idea of how DNA could be replicated. How could you make exact copies of the DNA? So the two strands of the DNA will separate, and a new copy will be made on each one of those two strands. Now, when we say that DNA is a hereditary material that stores information, and you can retrieve that information. So it's the only macromolecule that stores information in our cells. So if DNA is a storage molecule, that means the sequence of these nucleotides is the message, right? So your DNA contains functional units, and these functional units are called genes, right? Genes are physically and functionally functional units of hereditary, they are called genes. So genes are the units of DNA that can be transcribed into RNA, right? So when we say this is your DNA, it can be copied into new DNA. So we call that process replication, where DNA makes copies of itself. So the two strands of DNA will separate, and each strand will then make a new copy, right? So that's exact copy. But then there is another process where one of these strands is used as a template, and another strand is made that contains, instead of thymine, it contains uracil, and instead of deoxyribose is when this is deoxyribose. So carbon number 2 has a hydrogen instead of hydroxyl group. And in RNA, this is ribose. So both of them, they contain hydroxyl groups. Now, this RNA sequence is similar to this strand, which is we call it sense strand. This is antisense strand. So the antisense strand is used as a template for the formation of the RNA. So when this RNA is then decoded, so RNA is an intermediary that takes information from or in this case the messenger RNA is an intermediary that takes the information from the DNA and translates it into protein and passes it into protein. So this sequence of nucleotides, for example, CAU, UGC, CAG, these 3 nucleotides will encode one amino acid, right? So there are 64 different combinations that actually encode 20 different amino acids. So your DNA sequence is transcribed into RNA that is translated into proteins, right? So this is very important, that your sequence of DNA dictates what the sequence of your proteins will be. So why the sequence of the proteins is so important, we'll go to that in the next slide. But you know, this is called central dogma of molecular biology, and then there are exceptions to these. We know that there are enzymes that can make DNA from RNA, so there can be reverse transcription, but usually the flow of information is from DNA to RNA to the protein. Now, you know, just to reiterate, RNA is composed of ribose sugar. It has 4 bases: adenine, cytosine, guanine, uracil. It is single-stranded. DNA has deoxyribose. It has adenine, cytosine, guanine, thymine, and it is double-stranded. Now, when we talk about the sugars, as I mentioned before, this is your ribose sugar. So at the carbon number 2, there is a hydroxyl group. In the deoxyribose, at the carbon number 2, there is a hydrogen. When we talk about uracil and thymine, so there is a difference of this methyl group in both of these. So there are different, the uracil is only present in RNA, and thymine is only present in DNA. Now, when we talk about types of mRNA, so the RNA that I just showed you that makes protein is messenger RNA. We call it messenger RNA because it carries the message from the DNA to the protein, right? But there are many other types of RNA. Actually, the most abundant RNA is this one. 80% of the RNA in the cell is actually ribosomal RNA, right? mRNA's concentration changes all the time. It has a very high turnover rate. It's made, it's degraded. It's made, it's degraded, right? But the ribosomal RNA is the most abundant one. This makes the ribosomes that make the proteins. Similarly, you have transfer RNA. So these are the RNAs that take the amino acids, read the message on the messenger RNA, and then add those amino acids together with the ribosomal, जो ribosome है उसके साथ में, right? But then there are other types of RNA, microRNA, siRNA, long non-coding RNA that have, you know, plethora of functions, including, gene expression is the most obvious one, most studied function of these other types of RNA. But today's talk, we focus on the messenger RNA. Messenger RNA that carries message from the DNA to make the protein. Now, yes.
Yeah, so in one of the talks that was given, I think probably a couple of months ago, somebody was talking about small open-eating fields, so that lead to very, very small proteins that were recently approved. So would you be knowing about the RNAs that formulate those?
No, I have no idea. I know Khurram was working on these small proteins, but I don't know what kind of RNAs they.
Maybe mRNAs or?
If they are making proteins, we call them mRNAs. They can be shorter in size, but anything that carries the message is mRNA. Alright, so now, why proteins? You know, proteins are the most diverse macromolecule in our body. Diverse in terms of their function. They are, doing everything that you can imagine inside the cell apart from storing genetic information. So almost all the other functions proteins can actually do it. So I'll take you to few examples considering the diversity. I just mentioned that there are 20 amino acids that make up proteins. So to talk about the diversity, I can give you an example. If you make a dipeptide that contains 2 amino acids, that 20 to power 2, that's 400 different combinations. If you make a tripeptide that 20 to power 3, that's 8,000 different combinations. If you have a protein that has a sequence of 100 amino acids, that's 20 to power 100, that's 10 to power 130, right? That's, I read in a book that says that there are not enough sands grain of sand on this planet, or actually not even enough electrons as these combinations. And the proteins are usually bigger than 100 amino acids. There are proteins, there's a protein called titin, I think, that's about 27,000 amino acids. Some proteins are smaller, insulin is like 51 amino acids. So you can imagine the type of combinations, the type of molecules these 20 amino acids can make. And that is absolutely reflected in the diversity we see in the function of the proteins. For example, proteins can be enzymes. Most enzymes in our body, most of the machines that do the work in our body are actually proteins. There are some RNAs that are enzymes as well, but most of the enzymes are proteins. Similarly, proteins can be used to store. Amino acids can be utilized later. They are degraded in the lysosomes and then can be used to make new proteins. Proteins can be hormones. When you eat food, your pancreas releases insulin. That insulin helps take up the glucose in your blood by the liver cells, by the fat cells, by the muscle cells. Right? So proteins have very diverse. They can be signaling proteins, they can be motor proteins that help move my muscles. The way I'm speaking, I'm moving is because of proteins. Similarly, proteins can be defensive proteins. Antibodies, sabne suna hai antibodies, right? They fight infections. So antibodies are actually proteins. Similarly, transport proteins. Our cells have a boundary, that is lipid bilayer. That lipid bilayer doesn't allow, when you have a house, you don't allow anybody to come in, you have doors, right? Similarly, the cell has doors. These are transporters. The transporters are proteins that only allow certain molecules to pass through and don't allow the others to go through, right? Similarly, there are receptor proteins. Insulin goes and binds to a fat cell. Where does it bind? It binds to a receptor, a protein that it activates. Right? So receptors are also proteins. Similarly, there are structural proteins. आपके बाल जो हैं, आपके नाखून जो हैं, these are proteins. Right? Keratin, वो keratin लगाते हैं ना बालों में to make them, beautiful. So all these are proteins. So proteins have enormous functions, very important. These are the workhorses inside your cells. So when we say that DNA encodes proteins, that means if there is a change in the DNA, that change can or cannot be translated into a protein, change in a protein. वो cannot be इसलिए क्योंकि there are 20 amino acids but there are 64 codons. वो 4 into power 4 करेंगे तो 64 आ जाएगा ना आपके पास. तो जो आप combinations बना सकते हैं न्यूक्लियोटाइड्स के, they are 64, लेकिन जो amino acids हैं वो आपके पास 20. ठीक, तो कुछ कोडोन्स एक ही प्रोटीन को, एक ही अमीनो एसिड को कोड करते हैं, right? So sometimes a change in the DNA doesn't actually translate into change in the protein, right? तो क्या है कि mutations can be many different types. मैंने अभी आपको बताया normally क्या हो रहा है कि you have a DNA that's translated into mRNA, that is transcribed into mRNA, that is translated into a protein. If you have a mutation in the न्यूक्लियोटाइड में म्यूटेशन का मतलब अगर A है वहां पे G आ गया, या A है तो C आ गया, या A है तो वहां पे T आ गया. So these change in the nucleotides can change the sequence of the RNA because RNA uses DNA as a template. जब RNA बनता है तो DNA template use होता है. So if there is a change in the DNA, there is going to be change in the RNA. But sometimes the mutation doesn't translate into a different protein because it's a silent mutation, right? Because of the degeneracy of the code, genetic code key. However, other times a change in the DNA, when translated into or transcribed into change in the RNA, that can be translated into change in the protein. So structure of the protein, function of the protein can change as a result. So change in the DNA can result in the change in the structure or the function of the protein. So this is very important, right? So now many diseases, many genetic diseases, they are caused by changes in the DNA. Cystic fibrosis, genetic hereditary disease, है caused by a single amino acid change in the chloride transporter. वो change की वजह से वो transporter cannot is kicked out of the cell. वो cell कहता है कि ये पूरी तरह से fold नहीं हो रहा, so it kicks it out, right? So उसकी वजह से that disease is caused. Similarly, there are multiple diseases. आपका sickle cell anemia है, single amino acid change की वजह से the hemoglobin cannot carry enough oxygen. So wouldn't it be great if you could repair that change in the DNA, right? अगर एक mutative protein है that is caused by a change in the DNA, what if you add a correct copy of the DNA, right? That correct copy of the DNA will now make a correct version of the protein. A correct version of the protein will now do its function properly, right? Similarly, in cancer, we see that there are changes in the DNA. Some proteins are activated as a result of change in the amino acid, and some proteins are inactivated. We call them oncogenes or tumor suppressors, right? So this is the example of sickle cell anemia that I was talking about. A single nucleotide change can result in change in the amino acid, in the hemoglobin, and that actually even changes the shape of the cell, right? So as soon as our understanding of these genetic diseases and their why a change in the DNA is causing a disease, because it's causing dysfunction of a protein, right? So as soon as we started knowing that, people started thinking, oh, how can we repair that? How can we add a corrected version of that gene so that it makes a correct version of the protein, right? So this is where the gene therapies idea came from, that you can actually correct the incorrect version of a gene, right? So gene therapies idea is very old, right? You can directly inject the correct version of the gene in the body, or you can actually take the cells from a patient, correct those cells and reintroduce them. So it can be in vivo or it can be ex vivo, right? So depending on that. So gene therapy is a technique that modifies a person's gene to treat or cure a disease, right? But now we're talking about, here mRNA vaccines. That's what we are here for, right? So I'm coming to the point why, so what I've described to you up till now is why there is a need to put DNA or RNA inside the cells. Because we want to either vaccinate a person by putting a protein so that the cell's body's immune response produces antibodies, or we want to correct a gene that is mutated, right, that is causing the disease. So the first attempt to put RNA directly inside the mouse was in 1990 in this paper. So इसमें उन्होंने क्या किया कि they injected either the RNA, purified RNA, mRNA in this case, or the plasmid that encodes that RNA, right, into the mice. Mice के उन्होंने muscle के अंदर inject किया, and then they found कि actually जो RNA encoded protein expression है that was quicker. Within 18 hours they could see the protein that was translated from this mRNA, right, compared to 48 hours that the DNA encoded plasmid took. Similarly, they also showed that this was higher expression, right? So it was quicker and it was higher, but the take-home message was that RNA. उसके बाद for many years people tried to use RNA as a vaccine, but what happened? RNA produced an immune response. इम्यून रिस्पॉन्स क्यों है वो भी मैं अगली कुछ स्लाइड्स के अंदर आपको बताता हूँ. सो आरएनए people found was actually unsuitable. It was good, it was quick, directly translate हो जाता है foreign, you don't need transcription, right? If you could put it inside the cells, it will quickly make the protein, and that's what you want. You don't want के आप सारे genes को ठीक कर दें. अगर protein आपको मिल रही है body के अंदर which is functional, that's good enough, right? So अब क्या है कि the problem was कि high immunogenicity and low effectiveness. So उसको कैसे इलाज करेंगे कि आपका जो immune system है, we need to understand a little bit of our immune system before we understand कि why RNA is causing immunogenicity. Right? So our body contains two types of immune system. एक कौन कहते हैं innate immune system. So innate immune system is always there. Right? It's the first line of defense of our bodies against infection. मेरी skin is part of the innate immune system. आपकी आंखों में से आंसू निकलते हैं, they are part of the innate immune system because they contain antibacterial peptides. ठीक? तो आपका जो innate immune system है, that is quick, that is first response to the infections. ठीक? तो इस antigen non-specific, non-specific, इसको नहीं पता कि कौन सी किस्म का बैक्टीरिया है. इसको पता चल जाता है बैक्टीरिया है, वायरस है, ये पता चल जाता है, लेकिन ये नहीं पता कि कौन सी किस्म का बैक्टीरिया है, कौन सी किस्म का वायरस है. It recognizes special molecules, वो मैं आपको बताता हूँ. It has no memory. जो second line of defense है, that is adaptive immune system. It's antigen specific. सेल को पता है कि कौन से जो बैक्टीरिया है, कौन से प्रोटीन है, कौन सी वायरस की प्रोटीन है, उसके अगेंस्ट है, स्पेसिफिक है, and it has memory. एक दफा इन्फेक्शन होगा, दोबारा जब होगा, body will know कि this is the second time we are getting that infection. They will have cells that can produce antibodies to that, right? So vaccines का idea is from that activates the adaptive immune system. Now, the problem with the RNA is that actually activates the innate immune system. Why? वो इसलिए लोग शुरू के अंदर when the DNA was discovered, बैक्टीरिया में भी है, सारे ऑर्गेनिजम्स के अंदर है, ऐसे जेनेटिक मटेरियल, सब ऑर्गेनिज्म के अंदर जो है जेनेटिक मटेरियल डीएनए है. कुछ वायरसेस के अंदर आरएनए है, सिंगल स्ट्रैंडेड आरएनए, डबल स्ट्रैंडेड आरएनए, बट लेकिन जो भी प्रोकैरियोट्स, यूकैरियोट्स हैं, दे ऑल हैव डीएनए, एंड पीपल इनिशियली थॉट कि डीएनए इज सेम, बट देन लेटर दे फिगर्ड आउट कि कुछ मॉडिफिकेशंस हैं डीएनए के अंदर जो यूकैरियोट्स में, मेमल्स में है, प्रोकैरियोट्स में नहीं है. फॉर एग्जांपल, द सीपीजी डीएनए is methylated in us, but is unmethylated in bacteria. Right? So that's a change. So our body has evolved to detect the unmethylated DNA. For example, there is a receptor on our macrophages, on our dendritic cells called toll-like receptor 9. That recognizes unmethylated DNA. Right? So DNA या double-stranded RNA या single-stranded RNA या component of the bacterial cell wall या component of the virus, they are called pathogen-associated molecular patterns. क्या कहते हैं उनको? PAMs. Why we call them pathogen-associated molecular patterns? Because the cells, our immune cells from the innate immune system, the macrophages, the dendritic cells, they only recognize the pattern. The pattern क्या है? The pattern can be lipopolysaccharide. The pattern can be lipotecoic acid. The pattern can be single-stranded RNA. The pattern can be double-stranded RNA. It can be unmethylated DNA. Right? So, आपके पास you have these pathogen-associated molecular patterns. And one of those PAMs is nucleic acids. That includes RNA, that includes DNA. ठीक? Single-stranded RNA है, double-stranded RNA है, DNA है. And then there are receptors on the surface of our immune cells that recognize these PAMs. And those receptors, there are many different types. But one of the most important ones are called toll-like receptors. क्या कहते हैं उनको? Pattern recognition receptors. Right? And the most common of those pattern recognition receptors are toll-like receptors. But now, there are 11 different toll-like receptors. in humans 10 है, शायद mouse के अंदर 11 है, right? So these receptors, each one of them, it recognizes a particular PEM. For example, toll-like receptor 3 recognizes double-stranded RNA. Toll-like receptor 7 and 8, they recognize single-stranded RNA. Toll-like receptor 9 recognizes unmethylated CpG DNA. Toll-like receptor 4 recognizes lipopolysaccharide that is present on the cell wall of the gram-negative bacteria. Right? So there are similarly toll-like receptor 5. आपने flagella पढ़ा है ना? Bacteria have these threads that move them. Right? The flagella are made up of protein called flagellin. So toll-like receptors 5 recognizes flagellin. So all these toll-like receptors, they recognize specific pathogen-associated molecular patterns. Right? So what happens when they recognize these? So these toll-like receptors 3, 7, 8 and 9, they are actually present on the endosome. Endosome के ऊपर present. Why? When a bacteria or a virus enters the cell, it enters the cells through a process called endocytosis. Shabash. So endocytosis में से then it releases and goes into the, so that means it releases its RNA, its DNA inside the endosome. So these receptors, they recognize DNA or RNA inside the endosome. कुछ हैं जो जैसे लिपोपोलीसेक्राइड को toll-like receptor 2 है या other PAMs को toll-like receptor 1, 6 या 2 है, ये क्या करते हैं? They recognize at the surface of the cell. So what happens when the toll-like receptors, they recognize these PAMs? They initiate a signaling pathway that culminates in the production of cytokines. Interleukins and cytokines, and there are many these molecules that are produced that actually cause immunity and access. आपको एक example देता हूँ. Have you ever heard of septic shock? Some people who get infected by large amount of bacteria, gram-negative bacteria, endotoxin उसे कहते हैं. उसके जो cell wall के अंदर जो lipopolysaccharide है, that's also called endotoxin. So they actually die within couple of days because of the septic shock. So septic shock क्या है? That is lipopolysaccharide activating toll-like receptor 4 and that activating the cytokines. And if kappa B pathway activate होता है and that produces so much cytokines that your body actually can't tolerate that. So it actually dies. Okay? So the immune response through these toll-like receptors is very, very critical. So you don't want it to be excessively activated. Right? Now, अब क्या है कि जैसे मैंने आपको बताया कि the DNA is not all equal. जो आपके prokaryotes हैं, they have DNA that is unmethylated CpG. तो उसके अंदर है जो आपका हमारे अंदर DNA है उसके अंदर methylation. Similarly, the RNA is very different. Some of the RNAs, for example, ribosomal RNA, 25% ribosomal RNA modified है, उसकी जो bases हैं, they are modified. ठीक? तो ये जो modifications हैं, वो कौन सी हैं? I've listed few of these modifications. For example, 5-methyl cytosine है, so इसमें cytosine के ऊपर 5वें नंबर के ऊपर रिंग के ऊपर there is a methyl group. We have N7-methyl guanosine है, we have N1-methyl adenosine है और pseudouridine है. Pseudouridine, normal uridine में ये जो नाइट्रोजन है ये यहाँ पे होती है. So pseudouridine में ये नाइट्रोजन जो है that's on a different place. ठीक? Now these are present in, लोगों ने देखा कि actually this was known before. Nobody looked at that in that way, लेकिन it was known कि जो bacteria है, they have fewer of these RNA modifications. जो हमारे cells हैं, they have more of these RNA modification. Even अगर आप हमारे cells में से mitochondria निकाल लेंगे, तो mitochondria के जो encoded RNA है, they have fewer of these RNA modifications. लेकिन जो हमारा RNA है, that has more of these modifications, right? सो ये जो दो साइंटिस्ट हैं वाइसमैन और क्या नाम है कारिको, इन्होंने क्या किया, दे वर्कड ऑन ये फॉर एग्जांपल वही जो कि आपके आरएनए का स्ट्रक्चर जो मैं अभी आपको एज आई सेड आई मेड दिस टॉक फॉर वेरी एक्सेसिबल ऑडियंस, लेकिन आपके जो आरएनए है उसके अंदर यू हैव 5 प्राइम कैप है, जी कैप है, then there is a 5 prime untranslated region है, 3 prime untranslated region है, then there is an open reading frame है, and then you have poly A tail. Now all of these poly A tails हैं, 5 prime caps हैं, they actually regulate RNA की stability को भी regulate करते हैं, right? अब RNA stable होता है, इस तरह 5 prime UTR, 3 prime UTR, they also regulate stability among other functions, right? So when we talk about, अच्छा ये कि इन विट्रो अगर आप जब आरएनए डिस्कवर हुआ तो उसके सून आफ्टर लोगों ने ये भी डिस्कवर कर लिया कि यू कैन मेक आरएनए फ्रॉम डीएनए यूजिंग आरएनए पॉलीमरेज इन विट्रो इन द ट्यूब, राइट? सो यू कैन मेक आरएनए फ्रॉम अ पीस ऑफ डीएनए इनसाइड द ट्यूब, यू डोंट नीड अ सेल टू मेक द आरएनए, राइट? सो इसको हम कहते हैं इन विट्रो ट्रांसक्राइब्ड आरएनए, and in vitro transcribed RNA actually induces immune response. Why it induces immune response? That is the subject जो भी हम आगे डिस्कस करेंगे, ठीक? So ये मैंने डाला था कि जिन लोगों को पता नहीं है उनको बता दूंगा कि आपका not all of your gene has a coding region. So जब आपका RNA बनता है तो it's formed as a pre-mRNA. And that pre-mRNA is then processed. So उसके अंदर इंट्रॉन्स जो है वो रिमूव होंगे, उसके अंदर 5 प्राइम कैप लगेगी, 3 प्राइम poly A tail लगेगी, and this will be your mature messenger RNA, right? And this is like if you add the DNA fragment, usually a linearized DNA, plasmid लेके उसको linearize कर लेते हैं, उसको ऐड करेंगे, you add RNA polymerase, and then you add these nucleotides. And then appropriate buffer conditions में appropriate temperature के ऊपर you can make RNA in vitro. So this is called in vitro transcribed RNA. So आपने DNA से RNA जो है वो ट्यूब में बना लिया है instead of in the cells. So now I will talk about the 3 key discoveries that resulted in the award of the Nobel Prize. वो discoveries क्या है? पहला ये पेपर है I think in 2005. इसके अंदर क्या है कि suppression of RNA recognition by toll-like receptors, कि जो toll-like receptors हैं वो recognize करते हैं RNA को and they induce the immune response. So how can you suppress them? ठीक? तो इन्होंने क्या किया कि जो nucleoside modifications हैं उनको चेक किया कि whether they have an impact. उन्होंने hypothesize ये किया ना कि अगर बैक्टीरिया का डीएनए जो है उसके अंदर कम बैक्टीरिया का जो आरएनए है उसके अंदर कम मॉडिफिकेशंस हैं, हमारा आरएनए के अंदर ज्यादा मॉडिफिकेशन है, एंड आवर आरएनए इज नॉट रिकॉग्नाइज्ड बाय टोल लाइक रिसेप्टर्स, राइट? नहीं तो अपने ही आरएनए के अगेंस्ट हम रिस्पांस देते रहेंगे ना, राइट? सो देयर हैज टू बी अ डिफरेंस. तो दे एक्चुअली व्हाट दे पहला एक्सपेरिमेंट मैं आपको जो दिखा रहा हूं दैट एक्चुअली शोज कि इफ यू टेक आरएनए फ्रॉम बैक्टीरिया, इकोलाई से आपने आरएनए 2 डिफरेंट स्ट्रेन से आरएनए लिया, एंड देन यू टुक आरएनए फ्रॉम रैट, माउस एंड ह्यूमन सैंपल्स. आपने 293T सेल्स लिए. दीस 293T सेल्स डोंट हैव टोल लाइक रिसेप्टर्स. आपने क्या किया, उन सेल्स के अंदर आपने टोल लाइक रिसेप्टर 3 डाल दिया, राइट? टोल लाइक रिसेप्टर 3 विल रिकॉग्नाइज डबल स्ट्रैंडेड आरएनए, राइट? सो कंट्रोल के तौर पे उन्होंने पोली आईसी यहाँ पे मैंने काट दिया हुआ, लेकिन वो कंट्रोल इन्होंने डाला हुआ, लेकिन यू कैन सी कि जो बैक्टीरियल आरएनए है दैट इंड्यूसेस द IL-8 इंटरल्यूकिन के एक्सप्रेशन को, बट दीज आरएनएस फ्रॉम द रैट, माउस और ह्यूमन दे डोंट इंड्यूस दैट मच इंटरल्यूकिन. इसका मतलब है toll-like receptor is not activated by these RNA. Similarly, they did it for toll-like receptor 7. They did extensive study. I'm not going into detail. For example, they to ensure कि endogenous toll-like receptor नहीं है, they knock down toll-like receptor 3 and then they overexpress these and then they show कि only the bacterial RNA can induce the interleukin receptor, right? The second thing they did was कि they in vitro transcribed RNA. Now, when you in vitro transcribe RNA, you can add जो nucleotides हैं आप अपनी मर्जी से डाल सकते हैं. For example, you can add normal nucleotides or you can add the modified nucleotides. Modified कौन से वो भी मैंने बताया, जो आपका pseudouracil है वो डाल सकते हैं, जो 6-methyl adenine है वो आप डाल सकते हैं. So different जो nucleotides की modifications हैं वो आप you can incorporate into the in vitro transcribed RNA. So अब देखें ये जो एक्सपेरिमेंट इन्होंने किया ये मोनोसाइट ड्राइव डेंड्रिटिक सेल्स. So dendritic cells are very very important antigen presenting cells. ये क्या करते हैं, आप समझते हैं कि ये बीच में एक डेंड्रिटिक सेल खड़ा है, इस तरफ मेरे innate immunity है, इस तरफ मेरे adaptive immunity है. So they actually connect the innate immunity with the adaptive immunity. कि वो innate immunity को भी mediate कर रहे हैं, लेकिन साथ साथ वो क्या कर रहे हैं, they take the antigen present on their surface ताकि उसके ऊपर B cells आके antibodies बनाएं या T cells आके helper T cells आके आगे सारा कुछ करें, right? So आपके dendritic cells are very important modulators of immunity. तो इन dendritic cells में उन्होंने देखा कि ये देखें poly-IC क्या है, ये synthetic double stranded RNA, right? So देखें कि poly-IC जो है वो कर रहा है, ये और एक synthetic double stranded RNA वो भी कर रहा है. And when they take unmodified RNA, ये वाला नजर आ रहा है, और when they take the modified RNA, तो आप देख रहे हैं कि unmodified in vitro transcribed RNA induces TNF alpha, it induces IL-12. लेकिन when you add these modifications, it does not induce those वो जो आपका immune response है, cytokines को नहीं, TNF alpha भी नहीं कर रहा, IL-12 भी नहीं कर रहा, right? So that shows कि modification of the RNA can bypass the immune system. It no longer is recognized by the toll-like receptors. और इसमें जो टेक होम मैसेज है इस वाले पेपर में कि न्यूक्लियोसाइड मॉडिफिकेशंस, इसके बाद आई एम नॉट शोइंग, लेकिन उन्होंने डेंड्रिटिक सेल्स को बड़ा कैरेक्टराइज किया, देखा कि उनके सरफेस के ऊपर जो एक्सप्रेशन है डिफरेंट प्रोटीन्स का वो चेंज होता है या नहीं होता. तो जो आपके मॉडिफाइड न्यूक्लियोसाइड्स हैं दे डोंट एक्टिवेट द डेंड्रिटिक सेल्स. दैट्स द टेक होम मैसेज कि न्यूक्लियोसाइड मॉडिफिकेशन suppress the potential of RNA to activate dendritic cells. So dendritic cells are no longer activated. Now the second part is कि अब उन्होंने क्या किया कि देखा कि क्या RNA जो in vitro transcribed RNA है, whether it can be जो जिनके अंदर इन्होंने modifications की हैं, whether that can be translated or not. ये भी important है ना कि आपका immune system तो recognize नहीं कर रहा, but if your RNA that has these modifications in the nucleosides is not translated. So they did 3 main experiment जो मैं आपको दिखाऊंगा. पहले उन्होंने क्या किया कि in vitro देखा, ट्यूब के अंदर एक तो आपके पास in vitro transcription है, you can also do in vitro translation. दूसरा उन्होंने क्या देखा कि they saw in the cells कि cells के अंदर अगर हम RNA डालें तो does that RNA make protein. तीसरा उन्होंने क्या देखा कि क्या हम माइस के अंदर अगर इंजेक्ट करें RNA को तो क्या माइस के अंदर भी वो प्रोटीन बनाता है या नहीं. सो 3 चीजें. पहली क्या कि दे सॉ कि इन विट्रो अगर हम ट्रांसलेशन करें ट्यूब के अंदर तो अगर सूडोयूरेडिन है सूडोयूरेडिन है तो ये क्या करेगा कि आपका तकरीबन 2-fold इंक्रीज है इन द ट्रांसलेशन. ये इन विट्रो है, राइट. सो नॉर्मल आरएनए से ये मॉडिफाइड जो आरएनए है उसमें डबल द अमाउंट ऑफ ट्रांसलेशन होगी। दूसरा उन्होंने क्या दिखाया कि दे ट्रांसफेक्टेड 293T cells with this RNA and then they look for example मैं बीटा ग्लैक्टोसाइडेज का जीन डाल देता हूं, राइट, उसकी मैंने आरएनए बना लिया। दो तरह के आरएनए बनाए, एक आरएनए जिसके अंदर मैंने मॉडिफाइड ओलिगो न्यूक्लियोसाइड्स डाले, दूसरा जिसमें मैंने वाइल्ड टाइप ओलिगो न्यूक्लियोसाइड डाले, राइट। दोनों को मैं सेल्स के अंदर ट्रांसफेक्ट करता हूं। वो उनके ट्रांसफेक्ट इन्होंने लिपोफेक्शन से किए, लिपिड बेस्ड पार्टिकल्स बना के दे एडेड द आरएनए इनटू द सेल्स एंड देन दे लुक्ड। तो आप देखें कि जहां पे आपका यू है अनमॉडिफाइड है वहां पे कम है प्रोटीन, और जहां पे ये मॉडिफाइड है वहां पे आपकी प्रोटीन इज मोर। तीसरा उन्होंने क्या किया, उन्होंने माइस के अंदर करके देखा। माइस के अंदर दे वांटेड टू डू समथिंग कि जो माइस में प्रेजेंट नहीं है। फॉर एग्जांपल इसके अंदर उन्होंने लूसिफ्रेज के जीन को ट्रांसक्राइब करके विद और विदाउट न्यूक्लियोसाइड मॉडिफिकेशन दे इंट्रोड्यूस दैट इनटू द माइस एंड देन दे मेजर्ड द लूसिफ्रेज एक्टिविटी। ठीक, तो एंडोजिनस तो लूसिफ्रेज एक्टिविटी नहीं है ना माइस में, बिकॉज दे डोंट हैव द लूसिफ्रेज जीन। ठीक, तो उसके अंदर अगर आप देखें तो दिस इज द लूसिफ्रेज एक्टिविटी विद द जो व्हाइट आपको नजर आ रही है ट्राइंगल्स, दे हैव द लूसिफ्रेज एक्टिविटी इन द अनमॉडिफाइड RNA and the black dots are the luciferase activity in the modified RNA. So modified RNA has a higher activity, right? इसका मतलब है there is more protein of the same amount of RNA आपने डाला है, लेकिन एक में आपकी प्रोटीन थोड़ी बन रही है, एक में आपकी प्रोटीन जो है वो ज्यादा बन रही है। So take home message from this paper is that nucleoside modifications can enhance translational capacity while diminishing immunogenicity in vivo, right? Now the question obvious is कि what is causing the enhanced translation? कि ट्रांसलेशन क्यों हो रही है? दोनों तो mRNA है, आपने सिर्फ थोड़ी सी मॉडिफिकेशन की है, कुछ न्यूक्लियोसाइड्स को आपने मॉडिफाई किया। Why those cells are producing more protein compared to the cells that have wild type RNA, right? So what they did next was उनका जो तीसरा important paper, actually this is just from the Nobel Prize वाली साइड से मैंने लिया, just to show you कि आपका जो normal uridine है या जो आपका pseudo uridine है, so normal uridine will produce an inflammatory response, और जो आपका pseudo uridine है that will not produce an inflammatory response while both of them producing the protein. दोनों प्रोटीन प्रोड्यूस कर रहे हैं RNA, एक के अंदर यूरिडीन है, एक के अंदर सूडो यूरिडीन है। One of them is producing the immune response, the other one is not. The only difference between the two is the nucleoside modifications. So nucleoside modifications are not recognized by the toll-like receptors. ठीक? अब जो तीसरा इनका जो important paper है वो ये है कि why जो आपका modification वाला RNA, why is that producing more protein? Why is it translated better? तो वो इसमें मैं figures नहीं दिखा रहा है, made a kind of a cartoon. So अगर आपके पास RNA है, mRNA, if you put it in the cells, तो ये actually इन्होंने in vivo भी किया हुआ है. If you put it in the cells, that activate a protein kinase that is activated by the RNA is called PKR. ठीक, this actually recognizes the viral RNA. ठीक, ये PKR जो है वो क्या करेगी, वो एक्टिव होगी तो ये यूकैरियोटिक इनिशिएशन फैक्टर 2A को फास्फोरिलेट करेगी, उससे ये इसकी एक्टिविटी इनहिबिट हो जाएगी। सो इट मेक सेंस कि अगर वायरस का RNA आपके सेल के अंदर आ रहा है तो योर सेल्स हैव मेड अ मैकेनिज्म, वो क्या करेंगे कि उसकी ट्रांसलेशन को इनहिबिट करेंगे, राइट? तो ये क्या कर रहा है कि ये वायरस बाहर से जो आरएनए है जिसके ऊपर कोई मॉडिफिकेशन नहीं है, हाउ इट नोज कि ये वायरस ये आरएनए बाहर से आया, बिकॉज दैट आरएनए डज नॉट कंटेन मॉडिफिकेशंस। हमारे जो अपने आरएनए हैं दे कंटेन दीज न्यूक्लियोसाइड मॉडिफिकेशंस। सो ये जो मॉडिफिकेशन के बगैर आरएनए आया उसको इसने रिकॉग्नाइज किया, द पीकेआर गॉट एक्टिवेटेड, इट फॉस्फोरिलेटेड यूकैरियोटिक इनिशिएशन फैक्टर 2 अल्फा एंड दैट is required for the translation. तो आपकी ट्रांसलेशन थोड़ी कम होगी, राइट। नाउ इमेजिन कि जब आपका मॉडिफाइड सूडोयूरोडिन वाला आरएनए आपने डाला, तो दिस इज नॉट रिकॉग्नाइज्ड बाय द पीकेआर, सो पीकेआर कैन नॉट फॉस्फोरिलेट eukaryotic initiation factor 2 alpha, and the translation can go on smoothly. So that's why you have more RNA, more translation, more protein in this case than with the unmodified RNA. So this enabled, अब आपको दो चीजें पता चल गई कि modified RNA cannot produce immune response, and also modified RNA can actually produce more protein, right? So दोनों चीजें बेहतर हो गई ना, कि प्रोटीन भी ज्यादा हो गई और आपका इम्यून रिस्पॉन्स भी आना कम हो गया। So enhanced translation of mRNA with pseudouridine is mediated by reduced PKR activation. ठीक, तो आप PKR को एक्टिवेट नहीं करते, उससे आपकी ट्रांसलेशन जो है that is increased. Now this just is a figure that I added to show you that this was not the only work that was going on. There are lots of people doing lots of work, they cited them, and even after those discovery, even before COVID vaccine, there were other trials going on with the other vaccines, mRNA vaccines. So mRNA has been on people's radar for a long time, and the reason for that is it provided a very easy way. If you transfect viruses, आप ट्रांसड्यूस करें वायरस को किसी की बॉडी के अंदर, एक्चुअली जो 1999 में पहला जीन थेरेपी के ट्रायल्स हुए थे, उसके अंदर देयर वाज अ बॉय हु एक्चुअली डाइड फ्रॉम द इम्यून रिस्पांस ड्यू टू द वायरस। मतलब कि जीन थेरेपी से उसे कुछ नहीं हुआ, लेकिन जो वायरस जो जीन को लेके जा रहा था, दैट वायरस इंड्यूस्ड सो मच इम्यून रिस्पांस that patient actually died. और दूसरा क्या नुकसान हो सकता है कि when you put DNA inside our bodies, that DNA can go and integrate anywhere in your genome. वो किसी ऑन्कोजीन के ऊपर जाके इंटीग्रेट हो सकता है, right? And that can induce tumors. लेकिन your RNA is short half-life वाला RNA. इसकी half-life is very small. It can be degraded easily. सबसे ज्यादा जो लोग RNA isolate करते हैं, they can testify कि इसको तो आप उसके तरले मिनटे में करें तो स्टेबल नहीं रहता, right? So RNA की degradation is a challenge. तो इसका मतलब है कि RNA can't be integrated into the cells. So that's another advantage of using RNA as a vaccine. Right? So there are many different types of approaches that are used for vaccination. Vaccination क्या है कि you use a substance that can induce an immune response in a person, right? And that immune response has a memory. ठीक, तो next time when an infection strikes, that memory can actually attack that pathogen. So आपके पास बहुत सारी किस्मों के vaccines हैं. You can take attenuated viruses. Attenuated आप उनको कल्चर करते रहते हैं वायरसेस को until they become इतने docile हो जाते हैं कि they don't cause infection. उनको attenuated कहते हैं. Similarly, you can take viruses, उनको kill कर सकते हैं, बैक्टीरिया को kill कर सकते हैं, right? So there are, आप subunit vaccine बना सकते हैं. For example, you can take part of a protein of a virus or a bacteria, produce it, and then use that as an immunogen. Right? So there are many different types of vaccine. I'm not going into detail. लेकिन जो यहाँ पे वैक्सीन है, वो one type of vaccine is use of RNA और DNA. ये RNA और DNA को आप use कर सकते हैं, right? So why use RNA DNA? You use RNA और DNA to produce a protein. तो हमने कोविड के अंदर देखा कि दोनों तरह की वैक्सीन. There was Pfizer की वैक्सीन, Moderna की वैक्सीन was RNA वैक्सीन. एस्ट्राजेनिका की वैक्सीन या जो रशिया की वैक्सीन थी, they were both vector virus mediated vector transfer थी, DNA के transfer, right? So दोनों तरह की वैक्सीन. So this is just example आपको एक छोटा सा दिखाने के लिए कि कोविड की जो वैक्सीन है वो कैसे काम करती है. So you have this COVID virus that has a spike protein on its surface, and that spike protein is encoded by a piece of genetic material. उसके केस में RNA है. So what you do is you take that RNA, उसको DNA बनाएंगे, एम्पलीफाई करेंगे, इन विट्रो ट्रांसक्राइब करेंगे, एंड यू पैकेज दैट इनटू लिपिड लिपोसोम्स के अंदर, ठीक, लिपिड्स के अंदर आप पैकेज कर देते हैं, यू पुट इट इन अ पर्सन जो उनसे सेल्स को इन्फेक्ट करेगा, the RNA will go inside, it will be translated, it will produce the spike protein. When the cell dies, that spike protein is released, and that spike protein is taken up by these professional antigen presenting cells or called dendritic cells. So ये dendritic cells अपनी surface के ऊपर करेंगे, and that will help attract ये जो आपके T helper cells हैं, आपकी antibodies बनना शुरू हो जाएंगी, B cells will make those antibodies, and those antibodies will then attack the pathogens जो आपके viruses हैं उसको attack करेंगी, या if there is any infected cells, then another part of the immune system will actually cells हैं जो kill करेंगे, ठीक, उन cells को which are infected. So this is essentially the COVID vaccination, how it was made. Now, how do you produce drugs that are that utilize mRNA? So the first thing is you clone the part of the protein that you want to express, plasmid बना लिया, and then you linearize it. उसके अंदर you can make decision whether you include the poly A tail within your coding sequence या बाद में आप add करेंगे enzyme. So there are many strategies. So what you do is कि आप mRNA design करेंगे, आप mRNA को in vitro produce करेंगे, then you will select. There are many different types of. आपको हैरानगी होगी कि जो lipids हैं वो कोई आज से यूज नहीं हो रहे ड्रग्स के लिए, वो आज से 20 साल पहले भी ड्रग्स अप्रूव हुई हैं that use lipid as a carrier ट्रांसफर के लिए. वो कीमोथेरेप्यूटिक ड्रग्स हैं but they are being used for long period of time. So there are many different options available for encapsulating the RNA into these lipid particles. And then you do pharmacodynamic and safety evaluation. Pharmacodynamics क्या है कि if anything that drug does to our body is called pharmacodynamics. और pharmacokinetics क्या है कि whatever our body does to the drug. So pharmacokinetics क्या हो सकते हैं कि आप देख सकते हैं कि how long the RNA stays inside the cells, for example, or in a body, right? और pharmacodynamics क्या हो सकते हैं कि you can see कि whether it produces an immune response या it produces the protein, ये सारा कुछ आपकी pharmacodynamics हो सकता है. And then you do the safety evaluation. Safety evaluation you start it doing by first in small rodents, आप mice में करते हैं, rats में करते हैं, and then you go to the larger animal. You go to mini pigs, you go to dogs, you go to chimpanzees, and then eventually when that is done, you go to clinical trials. आप उसकी GCLP good clinical laboratory practice पे synthesis करते हैं, and then you take it to clinical trial. Clinical trial तीन तरह के हैं. जो phase 1 clinical trial है that just determines the dose of the drug. कि आप कितनी दे सकते हैं, कितनी जो है वो safe है. It determines the maximum tolerated dose or MTD. ये healthy individual में होता है, कोई भी इसके अंदर volunteer कर सकता है. इसके अंदर 50 के करीब 60 के करीब जो है वो patients होते हैं, इतने ज्यादा नहीं होते. And then you go to the phase 2 clinical trial. Phase 2 clinical trial is where you determine the efficacy of that drug. कि क्या ये जो vaccine हमने डाली है whether it's really protecting against the virus or not. ठीक, तो that is done in larger number, वो hundreds में होते हैं. And then you go to phase 3 clinical trial. Phase 3 clinical trial में आप क्या करते हैं? You look for long term safety of the drug, and also you look at whether the drug is better than the pre-existing treatment. So you compare them. This is multi-center study of 3000 patients के अंदर करते हैं, बहुत सारी जगह के अंदर करते हैं. जो कोविड के केस में it wasn't done for long enough. So FDA ने approval उसका emergency basis पे दे दिया था without an adequate longer safety उसकी करने के. तो safety concerns भी हैं, वो end पे मैं discuss करूँगा. But this is essentially जो जैसे आप करते हैं. तो आपकी जो applications है mRNA therapeutics की, वो apart from vaccines या infectious diseases के अलावा it can be genetic disorders, as I mentioned. आप gene therapy के लिए use कर सकते हैं. It can be cancer. कैंसर की ट्रीटमेंट के लिए आप कर सकते हैं. We know कि cancer is caused by mutations in genes that encode non-functional or overly functional proteins.
I have seen your project presentations now.
Sorry?
Seen your project presentations.
Oh yes, it's at 5?
Yeah.
Alright, so I'll just... Yeah, this last slide, Amir. So we have these, other applications. So you can make mRNA-based monoclonal antibodies. You can actually now clone the gene for the antibody, make RNA for that, and put that inside people. For example, कैंसर के लिए आप डाल सकते हैं HER2 एक वो है ऑन्कोजीन है that is expressed on the surface of breast cancer cells. उसको जो एंटीबॉडी टारगेट करती है वो herceptin है. People have actually done that for herceptin. Similarly, viruses का तो vaccines का तो मैं बता चुका हूँ. लेकिन you can do the mRNA based protein replacement therapy. वो मैं आपको बताया था कि जैसे cardiac problems के लिए you can do VEGF vascular endothelial growth factor या CFTR जो chloride channel है cystic fibrosis में उसको आप ऐड कर सकते हैं. And then gene editing के लिए भी आप mRNA को यूज कर सकते हैं. So challenges क्या है कि RNA is not stable. हमने देखा कि फाइजर की वैक्सीन जब पाकिस्तान में आई तो गवर्नमेंट को पता नहीं कितने -80s खरीदना पड़े. तो जगह जगह पे हर सेंटर के ऊपर -80s हैं. So उसकी वो बड़ा प्रॉब्लम है. आपके जो लिपिड नैनो पार्टिकल्स हैं उनकी सेफ्टी या बायोडिग्रेडेबल होने का या टिश्यू स्पेसिफिक टारगेट करने का उनको ये भी एक चैलेंज है. फॉर्मुलेशन उनकी कैसे कर सकते हैं. एक तो फॉर्मुलेशन है उनको बनाना, फिर उनको आपने इंजेक्शन देना है या कैसे देना है, दैट्स कॉल्ड फॉर्मुलेशन. इम्यूनोजेनिसिटी हमें पता नहीं कि कितनी लेंथ का, अगर लेंथ भी इफेक्ट करती है आरएनए की या नहीं करती, ये सारी चीजें. सो दीज आर स्मॉल चैलेंजेस बट ओवरऑल this has opened up so many fields कि आपको you take the DNA, transcribe it, put it in a lipid, and there you go. You have a drug that can replace a non-functional protein with a functional version of it. So thank you for your attention. I hope that it's
Thank you very much, Professor. Thank you. I don't think we have questions because we have to rush to next round of senior project presentation.
Next round of presentation.
Thank you very much, Professor. Very knowledgeable.